would like first to thank you so much, Jerry, because you were the first who switched on the light on the first tree genome, and you will give us also the way to switch on the light of thousands of trees everywhere in the world on different species. And thanks to these lights, we are really doing transformative change in breeding. So thank you so much. I would like in my talk just cover some context points of poplar breeding, then go to why genome information is of interest in breeder toolbox, then give you some transformative changes occurred in poplar breeding the last 20 years, and of course, because we have young scientists in the room, to discuss about expected further research efforts. Poplar breeding. Poplars cover about 62 million of hectares in the world. That represents 63% of fast-growing tree area, and this fast-growing tree area is expected of major use for our society. Genetic improvement was, is, and still be the most adapted strategy to meet all demands for both adaptive, quantitative, and qualitative performance. And why? Because vegetative propagation is easy, and we can really take value of individual variation, because clonal forestry has been adopted for decades, so society has been used to that. And then, because clonal selection provides high genetic gains and rapid deployment, the International Register of Clonal Cultivars is more than 400 entries with a lot of interspecific hybrid, and the process is continuing with more than 30 new entries every year. The current challenges for poplar breeding is really to remain poplar plantation and poplar natural forest, be able to maintain carbon storage sequestration. We need both use of forest. We need wood resources and the diversity of wood properties provided by poplar. Of course, Populus with its large genus is part of the biodiversity, and we need to keep it. And the last, we need also poplar to ensure land restoration, land protection. In the way of doing poplar breeding, it seems that, the breeding target change around. At the beginning, we talk about adaptation, but short adaptation. And in phases was given to growth, wood quality. But the carbon economy is more complex than single trait. Carbon economy, it's the link between all these traits. And adaptation in the context of climate change is increasing in importance, expected, increased resilience to abiotic and biotic risks. Regarding the genome information in interest for tree breeder, first, we start from genetic resources. And starting with a dozen of markers and now be able to use millions of markers change our lives. Regarding evaluation of performance, now we need to share new knowledge on the genetic basis of complex traits such as vulnerabilities and adaptive potential. That will help phenotypic prediction. Then, after evaluation, we have to process to selection and recombination, the cycle for breeder, and we need to adjust methods to accelerate and precise both selection and recombination. And at the last, it's not finished when you're a tree breeder. When tree breeder has to go into the field to see deployment. Deployment is the start of the use of this material. And we here see that we still need to use this information. Regarding genetic resources, the first most important information is that genomic information increase our knowledge of natural distribution on all different species. And population phenotypes are now known to be well predicted by genomic data as well as climate and geography. So that's something quite important for us to have this past history with us in the process of breeding. Regarding genetic resources, the breeder also is careful about relatedness on the material deployed, and so we have to compare. And you have on the left part, you know, the comparison of the distribution of the Populus nigra genitors in different European poplar breeding program in comparison to the natural variation. And you can see that, hopefully, in Europe, we succeed with complementary breeding program to cover and be used of all this diversity. On the right side is just to show you that, of course, a breeder is also providing some varieties for restoration of natural population and landscape. And thanks to this genomic information, we were able to build 2 multiclonal varieties, for example, to be used for river basin restoration in France, in Rhône, and these 2 multiclonal varieties are representative of a natural diversity in this population. Regarding evaluation of performance, first of all, you know, we have this first result regarding QTLs in large mopping pedigrees. And when geneticists say that what was important is to detect QTLs, breeders are not at the same way to interpret the result. They learn how to describe better their genitors. It's not really QTL informed, but the way for the first time they identified what are the potential of their genitors thanks to these genitors of large families. And the second one is to be able to explore the Mendelian sampling effects, because when you have full SIB, theory gives you the same level of relatedness between all full SIB, and thanks to genomic information, you explore much more. Regarding evaluation of performance, then we have this huge effort of combination of genome polymorphisms and even gene expression studies to detect genomic variant in large population through GWAS. And what's important in GWAS for breeders also, it's this capacity to detect adaptive signature in tree genome. Adaptive signature is the imprint of past natural selection, and tree breeders dream to be as efficient as natural selection. So to learn how signature is distributed could help breeders already to also have signature on the genomes. Here you have the results, show you signature on Charcot isomerase gene located on chromosome 10, and it's linked to contrary radial growth in Populus nigra. And you see here the signature of history in the eastern part of Europe on this signature. Last point, you know, when you have a large genetic variation to explore, sometimes it's in the rare events where you find very huge interest. And identifying rare individual, rare alleles is really difficult without this genomic information. So here you have an example of the detection of rare recessive alleles. So due to the nice collaboration with WOW teams. And it's just to tell you that we were then able to just catch the, something interesting with a low allele frequency in natural population, but most also a low frequency as individual, because homozygous genotype are still also very rare. So here it's to show you the power of this genomic information to really detect rich information. Regarding selection, individual sequencing, and high-density genotyping, really paved the way for genomic selection also in poplar. And what are the expected benefits? Of course, time gain, precision gain. What's it? I think as scientists, we need to be precise. Better management of genetic diversity, gain in selection intensity, but also maybe save some money to maybe invest more in diverse phenotyping. It's a way to balance and to change. Regarding the results, here is to show you that a proof of concept of efficiency of genomic selection, combining information on genome polymorphism and transcriptome in our Populus nigra breeding population. And you can see here that for 3 traits, height, circumference, and rust resistance, we have really multi-trait prediction efficiency. With the same set of S&Ps, you predict many traits. We are not limited, so multi-trait selection is really the most important focus. Regarding the use of molecular markers, the S&Ps from the genome, but also the transcript, show a better and higher level of prediction. And our breeding duration cycle was divided by 3. So genotyping costs now are not limited extra cost compared to phenotyping or archive cost. We really convince breeders, thanks to these other kind of course. The comparison, it's not to analyze the genotypic cost per se. Then, regarding deployment, just to say that it's important to facilitate traceability of natural and improved genetic resources at landscape scale and across country. Not only, we are, all these resources are international resources. And of course, the monitoring of gene flows between natural and cultivated stand. And this is a huge question, mainly raised by the society, gene flow. And surprisingly, in France, where people were afraid about this poplar plantation with exotic poplar species, they were not aware that, in fact, Populus nigra italica, which is from the same species of the natural population, in fact, is a more invasive with a high rate of introgression. So when you explain that to the society, that ornament is more dangerous than plantation, you stop any discussion. So further transformation are still needed to progress, to meet the challenge. We need new knowledge. Really, we need to identify and characterize rare genetic diversity to preserve it. It's time, even maybe it's too late. Genetic and epigenetic basis of plastic response. Plastic response is really to have a better view of short response because we have a very heterogeneous climate and it's important to have this short response. To identify genetic limits in key function response, and that the links with, these biological understanding and the role of functioning. And these limits, we must know them in extreme environment, and we need to know the limits regarding the allelic composition. Genomic options also have to resolve trade-offs because multi-trait selection is not so easy. We have a lot of trade-offs. Regarding transformation, we need open new tool. Open is important. We can see that we start in this integrative modeling. It's increasing the last few years, and we have to continue to do this more modeling work. To do that efficiently, we need open databases. Open databases to collect all information, genotypic, transcriptomic, epigenomics, phenotype at different level. And we really need to have this multi-source information connected. Then, and the last, for me, it's important that we continue to train and that to train differently tree breeding and genomics, and in order to update skills in genomics and breedings, and also to be able to integrate new jobs in our breeding sector to integrate this genomic technology. Thank you so much. I want to thank Jerry, all the actors of the international poplar breeding community, the Marcus Wallenberg Foundation to give me the possibility to be here to exchange with you. And thanks also all national, private and public funding institutions, and to thank the European Commission for their precious financial support to develop more efficient forestry breeding. We need them to continue to have lights.
Thank you so much, Catherine.